staff, please. And the recording is in progress, of course. So all I must say, it was before. Now I must say things more. Recording is in progress, please. Take care. Okay, have a nice time.
Thank you so much. We are here today. It was something planned from the beginning in B4EST project. Maybe is one point not changed by the COVID, hopefully. So we have succeeded with this important event at the final, at the end of the project. That's good. I would like to thank all contributors to these events. First of all, Isa, who tried also to welcome us in the best condition here, but also EIFC, which take in charge with all its team, all the organization, and because it's not easy to share presential and also connected things. So I hope all the connection will stay with us during the duration of this event. I would like also to thank a lot the scientific committee of this event. You know probably that we tried to build, and I would like to thank especially Santiago. Santi, I do not know where you are. Hello, Santi. Thanks, thanks to have coordinated that. And it was our choice to have a scientific committee composed with early career scientists who finally suffer from this COVID situation during the project. European project are the possibility for them to meet over teams everywhere, to work on challenges and scientific challenges of interest everywhere. And the situation was not happy for them. So thanks to all these early career scientists who accept to be in the scientific committee, who build the program, who will help us to have discussion during these 2 days and a half. So thanks a lot to you. I hope you will enjoy this event. And I hope also that this event will be the possibility for you to have your name put and unlight, because you need that for the future. You are the future for us. Then I would like to come also to thank all contributors of B4EST project for the progress done during the project, and with the possibility today to share the results produced. These results could be new knowledge, could be also new tools. We would like to share with the community, scientific community, but also community of stakeholders everywhere in order to build the future of forest. I would like to stick the point that in B4EST, our purpose was also not only to see ecological or technical breeding problem, but to include also the view of economic point of view, economic constraints, the way where we can meet with the view of economy in forest and the view of ecology in forest. And I think it's a challenge for tomorrow, where we have to meet. If we do not meet, there won't be any future for forest in Europe. So we need this meet, and I hope here are the result of the project. We could advance to continue the line to go on. So now it's time for me to let the floor to the people who want to share this new knowledge and new information to the scientific community. So please enjoy. Have nice days with us in Lisbon. Normalement, Santiago,
oui. Okay, so it's an honor to co-chair this session with Chetley, my figure, and yes, micro.
No, sit here because they are online.
For people to see you.
I have to sit there. Okay, so Chetley, want to. So can you hear me? It's okay. So well, I was saying that it's an honor to co-chair with my colleague Chetley this session. Just a very few words before starting with our keynote. So this session it's about different methodologies and aspects to improve breeding by using new methodological approaches and also the genomics tools. So we will illustrate with different examples how these new methodologies have been applied to our trees. And also when it comes to new methodologies in breeding, it's very nice to look around how other species are doing. And that's the role of our keynote speaker today, who is going to tell us a little bit about how they are doing in maize. So I don't know if Laurence, I see that you are there. Can you hear me?
I can you hear well, and you?
Yes, perfectly. So
okay, but so far I cannot share my screen. So.
Oh, you cannot share. Okay, but can we see the screen, the presentation on the big screen, or?
I did not send it, so I can send it, but.
Okay, well.
I thought I was going to share my screen, but it's not allowed. So what is the best that I send you my?
You can share. No, it's okay.
Okay, now it's okay. Try again if you can share your screen.
I still cannot share my screen.
Oh, okay. Well, otherwise,
it is said that the one who is organizing the meeting deactivated the screen sharing. So.
Okay, so is it set on? Did you activate the sharing or?
First, we prefer to send it to.
So it's better if I send my slide.
Yes.
Okay.
Yes,
okay, I will do it.
Yes, maybe if you can do it, maybe that would be the best thing. Yeah.
Okay, so I send it to Santiago. Is it okay?
Santé, it's okay. Or Benoit? Benoit, can you?
Benoit. Do I have the? Okay, Benoit.
The link for the Zoom meeting.
Yes, probably in your mail.
Yes, I will find it. Sorry for the time delay.
Yes.
I need to find it again. Sorry, I do not have it. Okay, I see it.
Okay. Yes, it's what she did. So did you find his mail?
Yeah.
Okay, very good.
No.
It was not that clear to. Okay.
Yeah, sorry about that. Yeah, but anyhow.
Did you receive it? It's a bit heavy. Sorry.
Okay, so he's checking. Yeah, he's checking. Electrons are traveling, so it's yeah.
So I won't be able to change my slides.
Yes. Otherwise, well, I know it's not very easy. Could you tell? Next we can do it. Is that okay?
Yes, but so far I do not see my screen, so I do not see what's on your screen. So
yes. Okay.
so it's a bit tricky,
Yeah, Yeah. So the only thing you see it's
I see a nice table with nice chairs.
okay, yeah. Otherwise, maybe we can reorient the webcam so that she can see the screen. Sorry. Reorient this
Oh yes.
webcam.
Oh yes. I don't know if you can do it manually.
T'as pas mis la bonne adresse.
Non, si tu remets ton adresse, t'as pas eu la bonne.
C'est pas la bonne?
Ah, il y a.
Ah, d'accord.
Voilà.
Ça qui est un peu
Si,
embêtant.
si, elle verra. Elle verra les fonds.
Alors là, j'ai pas au point. Il faut. Ouais.
Laurence, well, we have just got your so maybe if you put it in the yes.
Yes, the system does not allow.
Laurence, can you see the screen now?
Yes, I can see it. Yeah, it's much better. Yeah.
It is not optimal. I'm sorry about that, but anyhow, so if it is okay for you, then you can tell us when to change the slide.
Okay.
Okay, so the floor is yours. Yeah.
Okay, so thanks a lot, Leopoldo. So first of all, so I would like to thank the organizers for inviting me to be here. I'm so sorry not to be in Lisbon indeed, because I would have liked a lot to be with among you. I know that Lisbon is a very nice city, and it would have been a nice opportunity to discuss with your community. So today I'm going to present you what is done in terms of genomic selection in an exotic species compared to yours. I will talk about maize and how to use genomic selection more specifically to better manage and enrich genetic diversity in breeding programs. And I have to say that this work was mostly done, of course, by PhD students. So Alizarine Lorenzi, Adam Assay, but most of all, Antoine Allier. Who did his PhD with us, supervised mostly by Alain Charcot and myself, and also other colleagues. So next, please. So maybe most of you are not really familiar with maize. So just to remind you that maize was domesticated indeed in Mexico. And then it was adapted to different climates and most of all temperate climates. As you may know, it is an analogous species that exhibits a strong heterosis effect. And so this was valorized in the 50s by a clear revolution in terms of breeding, because breeders, they move from landraces, populations of heterogeneous plants, to single cross hybrids. And so this led to the formation of what we call heterotic groups, that is to say groups that were complementary to each other in order to optimize the value of intergroup hybrids. And this has also several consequences. So first, you can see on this graph in the middle that the grain yield increased a lot after moving to hybrid breeding. It also generated a very competitive private sector. And something that is important is that inbred lines that are developed by breeders are not available to the other ones. Unless inbred lines are protected using patents, and then inbred lines become available when the patent expires. And so this has, as a consequence, the old population varieties that have been used until the 50s, they can be considered as sources of genetic diversity that were only partially exploited when the breeding moved to hybrids. And the issue is that it's difficult to use them now because of the large performance gap with the elite material that we have today. So next, please. Okay, so in terms of breeding scheme, so the selection is a reciprocal recurrent selection scheme. That is to say that the selection is done within each heterotic group. So in each heterotic group, inbred lines are crossed in order to produce new progenies. And now, thanks to the use of haplodiploidization, breeders, they generate hundreds of new inbred lines from these crosses. And these inbred lines are not selected for their per se value, but they are selected for their ability to give good hybrids. And because it is not possible to test all the possible hybrid combination to facilitate the selection, the candidate lines are first crossed to a limited number of lines from the opposite group, which are called testers. And then the hybrids are evaluated in order to select for the general combining abilities of the inbred lines. Then only the best ones are crossed using a partial factorial design in order to identify the best hybrids. And then the selection at this stage is done on the GCA, but also on the specific combining abilities. So next, please. No, yeah. So maybe it's you may found that it's not that long, but I would say that it is nevertheless a long process because even for an annual species like maize, it takes at least 8 years or 5 years to obtain a new variety. And it is also an expensive breeding scheme because of these different stages of phenotyping that we have. And also regarding heterosis, in fact, this breeding scheme only partially exploits the complementarity between heterotic groups. So next, please. Yes, so as for many other species, I mean, clearly the advent of genomic selection can be considered as a breakthrough innovation in maize breeding. So here I recall you the basic principle of genomic selection. So the idea is that we are using a population which is called a training population in which individuals are genotypes and phenotypes. Using both types of information, it is possible to calibrate a prediction model and to use then this prediction model in order to predict the value of any new individual using only its genotype, so its marker genotypes. And of course, it opens a lot of possibilities to select individuals by performing some early selection, also to reduce the amount of phenotyping in the breeding schemes, select also for traits that cannot be evaluated on all the candidates and so on. So in this part, I mean, the key main point is, of course, to define a good training population. So we have worked on this topic, but I will not talk about that today. So next slide. So what I want to mention to you is that genomic selection was really adopted really soon, indeed, in maize breeding. Because quite quickly, so the first publication on genomic selection can be attributed to Mavison in 2001. And you can see here on the upper part of my slide that in 2003, there were already some examples of the use of genomic prediction equations in maize. And this was done here in this case in Monsanto breeding company, where they tested this application in a biparental population using a very limited number of markers, and they were able to find that it was advantageous compared to other previous marker-assisted selection techniques. But clearly, the beginning of genomic selection in maize started in 2009 after maize was sequenced. And because it, of course, facilitates the development of some genotyping arrays. So first in the private sector and then in the public sector. And so today, the genotyping cost in maize, they can be considered as being equivalent of one feed plot. So clearly, genotyping individual is really competitive. And so I think that we can consider that genomic selection has now been adopted by most, if not all, maize breeding companies. And this can be also illustrated by the large number of publications per year on the use of genomic selection in maize. So next, please. Yeah, so in a breeding scheme, genomic selection can be used to facilitate the screening of candidates by avoiding to phenotype all the individuals, which is a way to increase the selection intensity and also to complement the information given by the phenotype. So it's also a way to increase the accuracy of the prediction. One advantage also of using genomic selection in maize was that it led breeders to consider more globally the genetic variability that is available in their breeding scheme, which was not the case before because they did not use pedigree information, for instance. Really, the main application of GS is to perform what is called the sparse testing, so to remove part of the phenotyping. So next, please. Some of the breeders also use genomic selection to perform some rapid recycling of diversity without any phenotyping, but I would say that this is not the common practice to all the breeders so far as I know what they are doing. So next slide. Something that can be done also with genomic selection in maize is to clearly use it to revisit the reciprocal selection scheme. And what has been tested was to replace indeed these tester evaluations that I showed you before by some genomic predictions that can be calibrated directly on hybrids that can be generated using incomplete factorial design between candidates before being selected. And this has several advantages. So first is it avoids this stage of phenotyping that is done separately in two groups. So it can be a way to reduce the number of hybrids that needs to be evaluated. And so to increase the number of candidate lines that can be evaluated at a fixed number of phenotype hybrids. And potentially also it offers the possibility to select directly on the GCA and SCA component. So this strategy has been tested in the framework of a project that we have with Pro-Maize Association, so an association of maize breeders. And so within this context, we perform some simulations and we also perform some experimental evaluation of this strategy. And we were able to find that for traits that indeed show some specific combining ability, using a factorial instead of a tester design can be really beneficial in terms of prediction accuracy. And this was validated experimentally on the right for dry matter yield in this experiment that you see. I do not want to spend too much time on this, but just to say that this approach indeed may strongly modify the breeding practices by removing this stage of evaluation on tester progeny. And so this is currently tested by several breeding companies. Yes, you can move to the next. But one key issue in maize breeding is also the genetic diversity, because as I told you, so genetic diversity is structured in heterotic pools, and in each pool, the effective number of individuals is of course not that high, which increases the risk indeed of diversity loss due to drift and to selection. So in order to evaluate the decrease in diversity that we may have in breeding programs, we had a collaboration with a breeding company, so Heritability, in the framework of a PhD of Antoine Allier. And what Antoine Allier did is that he evaluated the genetic gain that was achieved in this company in two heterotic pools, the dent and the flint. And you can see on the left that along the years, he clearly saw an increase in the genetic value of the two populations, but he also observed a decrease in terms of genetic variability that you can see on the right, which was really strong in the dent pool, which was not that strong in the flint pool, but you can see that there are 2 points that are a bit different from the others, and they correspond to years where external diversity was introduced in the program. Something that is nice also with markers is that it is possible to look at the diversity along the genome. And you can see on the bottom of my slide that we were able to identify large regions where the diversity was very low and which were near to fixation. You can see here, for instance, on chromosome 4. So clearly there is an issue in terms of genetic diversity in these programs. And we can ask the question how to better use the diversity in breeding and how to best integrate new sources of diversity. So next. Yes, and this is really a clear issue, and I know that you are all concerned by this, and it is also the case in maize. We know that there are now key challenges related to food security and the fact that we have to face some climatic change and also changes in terms of societal demands. There is a constantly rising demands for plant-based products. And clearly we need more accurate and faster breeding methods that can be easily adapted to new and changing targets. So in this context, we can see genomic prediction as genomic selection as a key opportunity. But we also know that recurrent truncation genomic selection in close breeding programs could rapidly erode the genetic diversity and thus limit the long-term genetic gain and the breeding sustainability. And so the breeders, they clearly need to efficiently convert the diversity that they have in their program into genetic gain, but while keeping a decent level of neutral diversity. And there is also a need to anticipate the farmer needs by introducing constantly external sources of favorable alleles coming from the genetic resources to efficiently sustain the breeding progress. And so in these two aspects, we believe that genomic selection can be helpful. So next, please. So when breeders have to decide which crosses they are going to make for the next generation, they need to select among all the potential crosses the best one. And the value of a cross can be defined by the average value of the two parents that are crossed. And this part is easily predicted using genomic prediction. It corresponds to the mean of the parental breeding values. And but what we really want to predict is indeed the value of the best fraction of the progeny that we can select from this cross. And this corresponds to the best individuals that are here with the red box. And this corresponds to what was called the usefulness criterion. And in order to predict in advance what will be the usefulness criterion of a cross, we need to be able to predict the within family variances. And this can be done also using markers. And so next. And so Antoine Allier proposed indeed to select crosses based on what he calls the UCPC criterion. So what is the UCPC? It is a combination of the usefulness criterion. So using markers and taking into account the probability to recombine and the linkage prions that he has and the genotype of the two parents, it is possible to predict with the marker effect the value of the best progeny that can be derived from the cross. At the same time, we know that in the progeny of the two parents, the inbreed lines that can be derived, there will be a mixture of the two parental lines, some being more like P1, some being more like P2. And it is possible, once again, to predict this and to see the contribution of each parental line as a trait which is correlated with the value, with the performance of the individuals, as you can see on the graph. Next, please. Using marker, we are able to predict the distribution of the performance, the parental contribution of the progeny among the individuals that are going to be selected that are within my red box. And so next. And so this is for a given cross, but of course, if we want to maintain diversity over the breeding program, it is important to take into account the complementarity between crosses, with the idea to balance between the short-term response to selection and also the loss of diversity that the crosses choice may generate. And so to do that, Antoine proposed to use an adaptation to the optimal cross selection using the UCPC and using a differential evolution algorithm to find the list of crosses that will maximize the expected performance of the individuals and that will also under the constraint of a reduce decrease in terms of diversity that can be estimated genome-wide using markers and the relationship between the individuals and the percentage of each parental lines that will be kept after selection. And so, of course, we expect diversity to decrease over time, and we can constrain this loss of diversity using different equations. And here, what he considered was he accepted a linear decline of the genetic diversity over time, so the green curve. So next, please. So what he did is that he compared using simulation the genetic gains that can be achieved with different selection strategy. So in black, you have a truncation selection using only the parental mean to select the crosses. And you can see that using the usefulness criterion, so in blue, is already a way to increase genetic gain compared to what can be done using only parental mean. And it reduces also the loss of diversity along generation, as you can see on the right. So next, yes. So using the conventional optimal cross selection, is a way to improve the efficiency of selection in the long term, as you can see in orange here. And using the adaptation with the UCPC can lead to a further increase, a slight increase of the genetic gain. And what is important to see is that we have a long-term gain in terms of efficiency and only a short-term penalty, as you can see at the beginning of the graph at early generation. And this is achieved thanks to a better management of the genetic diversity, as you can see on the right. Okay, so nevertheless, even if we can, to some extent, control the loss of diversity. I mean, the loss of diversity is, of course, something that we cannot prevent unless new sources of diversity are introduced in the breeding program. And so, so the key point is how to identify donors that can be used to enrich a gene pool. And in maize, it is really tricky because, as I told you, we do not have an easy access to the diversity which is available from competitors. And there is a large performance gap between the the land races and between the elite material. So one way to identify donors was tested using data that were generated within the Amazing project. So in this Amazing project, which was done in partnership between private companies and public sectors, a panel of diversity was assembled, and it corresponds to inbred lines coming from different cycles of selection from the public sectors, but also from the private sector with more recent inbred lines. And so this panel is a kind of link between genetic resources and elite material. And so what Antoine Allier proposed was to use this panel as a training set in order to estimate marker effect, and to use then the marker effect in order to predict the value of potential donors that are included or not in the panel, and also to evaluate the complementarity between the donor and the elite, and so to select the best donors. So next. So he experimentally tested the possibility to really do some prediction of what is going on in elite material using such type of collaborative panel. So here you have the results of the prediction accuracy that was found in the elite material of Heritability Society. So I cannot say that the prediction accuracy was very high, but nevertheless, it was not ridiculous. We obtained a prediction accuracy of 0.4. And what is interesting to note is that for a trait like yield, we were able to, in a way, predict the evolution of the genetic gain along generation. You can see that the most recent inbred lines are predicted by genomic prediction as being better than the oldest one, which are in blue. And so if genomic predictions can predict in a way the selection tendency that occurred, we can see that maybe it can be used to identify indeed interesting sources of diversity that will contribute to the next to come genetic gain. So next. So how to identify donors that can best complement inbred lines. So the idea was to use allelic effect estimated using genomic prediction. So when you do that, you have a lot of markers, a lot of additive effects that can be estimated at each marker. So it's a bit noisy. And the idea is to consider some sliding windows and to average the effect of the markers over the sliding windows. And then to compare the alleles that are carried out by different here inbred lines, so lines B and lines A. So in this example, we can see that at the beginning of the chromosome, the two lines, they are similar, they have the same alleles, but then they are different at the end of the chromosome, and we can identify chromosome regions in which line A is better, carried better alleles on average than line B, and it is the opposite in the chromosome region next to it. And so using this type of information and also a kind of iterative process, we can choose the best donor lines that would best complement an existing pool, for instance, some elite inbred lines. And so this was done by Heritability. And so using the Amazing DEN panels, they were able to identify some inbred lines that best complement the 3 elite lines that you have here in blue, and also to use the UCPC to identify the best crosses to be made using these donors and these elite lines. So we do not have the evaluation of the value of these crosses, but in order to better document and also to see how to best integrate donors in breeding schemes, so Antoine Allier performed some simulations. And so in maize, there are different types of genetic resources that can be used. So we can use recent improved resources. So in bread lines from another breeding program from the same company, for instance. So this may correspond to the red curve with recent material. We can also use some older material, for instance, in bread lines that has been released, so XPVPs. And we can also try to introduce directly genetic resources from landraces. And what you can see here is that if you do not introduce any diversity, so after a given time, you will reach a plateau. So using recent improved donor lines is clearly beneficial. You can see here in red. Older improved breeding lines or older genetic resources can be beneficial, but it's not the case if the genetic resources are really old. And also you can see that there is really a big short-term penalty that occur in this strategy. So in order to overcome this, so please next slide. What proposed Antoine was to use a bridging generation between the genetic resources and the elite compartment. And he showed here, so you can see here with the dot lines, that using this bridging generation was a way to reduce the short-term penalty and to observe a genetic gain even when introducing genetic resources that are really old compared to the elite material. So next, please. So just to conclude, so take-home messages. So in case of maize, so genomic selection, as I illustrated, can be used at different stages, and it can be also used to potentially revisit the whole breeding stage. In terms of diversity management, it clearly provides also some opportunities in order to facilitate the control of the diversity within the elite compartment, but also to facilitate the use of genetic resources. So first, by identifying among the genetic resources those that are complementary to the elite germplasm. Genomic selection can be used to improve the genetic resources. I did not illustrate that, but of course, it can be used to reduce the performance gap between the elite and the genetic resources. And it can be used efficiently to better manage the introduction of the donor line into the elite germplasm while maintaining the genetic diversity constant in the elite compartment. So next. So in terms of prospect, clearly topics related to diversity are something that on which we are working a lot today, because it's something that is also well adapted to private public partnerships. Private breeders, they cannot do that alone because it is a lot of investment. And I mean, in the public sector, we know genetic resources a lot, and we have worked a lot on this topic. So there is presently an experimental participatory program of introduction of diversity, which is going on in the framework of the Pro-Maize Association. It is a PhD of Dimitri Sanchez. There is clearly a need to further characterize the genetic resources in the case of maize, and more specifically the landraces. So there are several projects that are going on, and we are collaborating on this with Brigitte Guénard in Montpellier. So several projects aim at extracting some DH line from landraces to facilitate their conservation and their use in elite germplasm. There is a project by Sarah Ben Sadoun, and also a main aspect is to increase the genotyping of landraces and also their phenotyping in order to identify original sources of diversity that have been underused so far, and to use genomic selection and GWAS to identify among these genetic resources adaptative QTLs and also predict not yet phenotype landraces. And so the idea is to combine different types of information that you may have regarding these landraces in order to achieve that. And we have some preliminary results that shows that it can be really interesting. And so with that, I would like to thank you for your attention and to thank all the researchers and also technicians from our lab who contributed to this work. Of course, the different PhD students that are in our group. I would like I would like to thank also external collaborators from INRAE, but also for other institutes, and more particularly people from RAGT, so Christina Lehermeyer and Simon Tessèdre, who supervise the PhD of Antoine Allier. And next, please. Okay, and also all the partners of the ProMaize and the amazing project. And thank you for your attention. I will be happy to take questions if you have any. Do you hear me?
Il n'y a pas de, ça marche pas.
I hear you.
Yeah, we have time maybe for a very few questions, if the micro works. Okay, so well, is there any question? Okay, I have a yes, Catherine.
I have a question to you, Laurent. Thanks so much. I was impressed to see how close we are with the different challenges. You have on maize and some challenges. We have also forest breeding regarding both the complementation in hybrid production, but also this concern of maintaining genetic diversity, make the use of more favorable allele coming from a wider genetic resources, not so much explored in terms of performance. My question is, I do not see in your talk a specific thing regarding the multi-trait approach. So we are all concerned by genomic selection with multi-trait approach. Do you see that genomic selection could help us to better manage some possible trade-off or limitation due to this multi-trait challenges, or what is your opinion on that?
I think it's yes, it's a very interesting question. To be honest, so far we did not that much work on this topic. But of course, I mean, all the approaches that we developed, they can be extended also to handle some multi-trait prediction concern, because with genomic predictions, we can also predict not only what's going on in terms of variability for a given trait, but we can also consider jointly several traits, and so predict in a way the correlation between the trait and the evolution of the correlation after selection. So of course, I think it's really something interesting to do. What we believe also is that, of course, selection is not done on a single trait. It is done on multiple traits, and sometimes with, and quite often with some negative correlation between traits. And so, yes, I am quite sure that genomic prediction can be a way to better handle this. And also to take advantage of the fact that some traits are more easily predicted than others. And so they can be helpful to improve the prediction accuracy of traits for which it's difficult to predict, and for which, for instance, the phenotyping is really really expensive and cannot be done at a larger scale. So I did not present results on this topic, but yes, I do believe that that it may be an opportunity also for genomic selection for this trait. And I would just want to add that, because now we are moving more, I would say, to to see what could be the interest of genomic selection in more, I mean, not only in the elite germplasm in maize, but also in land races where we have some populations to handle, where we have some individuals that are not fixed and so on. I think that in terms of method and strategy, we are also closer to what is done currently in perennial plants and in forest species. So I think there are room for improved collaboration between the different species at this point. Yeah.
Okay. A very short question before next presentation. Laurence, I have a very short question. Yes, in your comparison between factorials and testers, there are very classical papers saying that factorials could have a role in managing genetic diversity. Did you see that in your simulations or in your experiments?
Not really, I have to say, because diversity was not the main concern here. So no, sorry, that's something that we didn't look at. But yes, that's a good point. Yeah.
Okay. Yeah. Well, thank you very much for your presentation.
And thank you again for your invitation. And so I wish you all a nice meeting.
Okay, so okay, so we can now start with our first speaker, Victor Papin, who will talk about reactions norm for maritime pine. I think the best thing you sit there.
It's working? No. Yeah, we need to. I need to get really close. Yeah, that's the only issue.
So hello everyone, my name is Victor Papin. I'm a PhD student working on genomic selection of maritime pine at the INRAE of Pierreton in France. I'm supervised by Laurence Moreau from Pierreton and Leopoldo Sanchez from Orléans. And today I will present you a part of my work about the construction of correction for maritime pine. So to begin with, just a few elements of context about the maritime pine breeding in France. So the land forest is the biggest plantation forest in France. It represents over 0.8 million of hectares and around 24% of the French wood others. So it's a very intensive production area. Maritime pine wood is mostly used in carpentry, joinery, and stationery. And in the case of the land forest, almost all the plantations are done with improved seedlings based on the local problems. So the maritime pine breeding started during the 60s and follow a recurrent selection system that reached today the third generation. This program is mainly based on growth rates like height, circumference, or straightness, but it also evaluates adaptation to the environment and wood quality. And in this program, genetic value are only estimated with pedigree. Molecular markers for the moment are only used to do pedigree reconstruction. So we can see in this context that the classical selection mostly focused on final growth rates. These parameters sum up the tree's growth, so they are very integrated and do not take into account the phenotypic evolution across the year facing the change in environments. So in order to explicitly take into account this environmental effect and to work with more dynamics or more explicative traits, we decided to model norms of reaction A norm of reaction is a pattern of phenotypic expression of a single genotype across a range of environments. And it will allow us to estimate genetic value, not only for the final trait, but for the whole growth model facing a change in environments. So the statistical tool that we are going to use is the random regression models. This kind of model integrates three types of data: the phenotypic measurements of the trait whose evaluation we want to model, this index variable, which is an explicative variable, in our case, a climatic or environmental variable, and then the genetic data to take into account the relationship between individuals. So these three types of data are integrated in random regression in order to model the phenotypic evolution across the index variable and to extract the genetic part. So I will detail today each part of the process, starting with the data. And first of all, the phenotypic measurements that we have for this study. So we have an experimental design composed of two locations in Cestas and Escos in the southwest of France. These two plantations have been made in 1996, so we have 26 years old trees. And each location, we have 150 half-seed families with 35 individuals in each family. So for all these individuals, we have classical growth measurements like height or circumference, but we also have a core sampling. So among the 150 available half-seed families, 25 of them were selected as representative of the phenotypic diversity. And in each family, 13 individuals have been core, so as shown on the image. So we have around 300 individual core in each location, and we'll focus on this phenotypic data to build norm of reaction. Just to finish, we also have annual climate measurements to characterize each one of our locations. So as I say, we will focus on the core sampling. So after the field work, the cores have been cut into balls and then X-ray to get this kind of profile at the end. So this profile represents the evolution of density from the tree pith to the bark. Each red line delimits a ring, a year of growth. And for each individual, we can summarize the annual growth by general parameters, like the ring width, ring surface, or ring mean density. And as we have one profile for per individual, we can at the end build the and plot the mean trajectory of the whole population across the year. So that's what's represented here. We have the ring width, ring surface, and ring mean density across the year. So in red, it's the mean trajectory for the 300 individuals of Cestas, and in blue, the same for the 300 individuals of Escos. As you can see, ring width and ring mean density seems very influenced by the year, more precisely by the age of the trees. For ring surface, after several years of intense growth, we reach a kind of plateau with important inter-annual variation. So for the rest of this presentation, we will focus on the ring surface, which is easier to model in the first time. And we decide to focus on study range from 2004 to 2019, where we can consider the age effects as negligible. So this is the trajectories that we are going to model, but before we need to replace the x-axis by an environmental variable, our index variable. So that's I'm going to detail you. So first you have the evolution, the mean evolution of ring surface across the year. So it's for the 600 individual, it's just the same as before, just all the individuals are grouped. And to build our index variable, we decide to characterize each year by its global level of aridity through the Merton index. So this index is calculated as a ratio between the cumulative rainfall and the mean temperature. Actually, it's calculated for each month, and we average over a period from March to October, which corresponds to the growing period of maritime time. So at the end, we have one level of aridity for each year. This level of aridity can be very different considering the year. So it's represented in the second figure. We can see that we have very humid years until semi-arid years. So it constitutes as many different environments controlled by the tree during its lifelong growth. And so at the end, we take the same as the first picture, just we replace each year by its global level of aridity and reorganize it from the driest one to the most humid. So it modifies the shape of the curve, as you can see, and came close to the biological expectations that we could have, namely reduce growth, reduce ring surface in driest environments, and it tend to increase and reach kind of maximum in most humid environments. So this is the final trajectory that we are going to model with random regression. Just before we need the third and last type of data, which are the genetic data. So to characterize the relationship between individuals, we have pedigree information that can help us to build a matrix represented here. And also all our individuals have been genotyped, so we can build G matrix represented here. So in both cases, we find the alcyl family structure with 25 small blocks across the diagonals. But we also show that with genomic data, we enhance and precise some relationships that were unknown or ignored with pedigree. Here it's the same idea. It's a representation of the relatedness calculated with pedigree information or with genomic markers information. We can see that for each level of pedigree, the genomic relatedness calculated is centered on the theoretical value, but we have a quite important range of variation that allow to precise the relationship, or for example, to identify some pedigrees error. So once we have these three types of data, we can then launch our random regression model. So this model, so we want to model individual trajectory across our different levels of aridity. So our phenotypic trait will be the ring surface of all individuals in all environments. And then our model is split in two parts, a fixed part and a random part. In the fixed part, we have one element, which is the mean trajectory of the whole population. So this trajectory is considered as the fixed effect in the model. In fact, we are a little bit more precise. We decided to model two mean trajectory, one for each location, one for Cestas and one for Escos. And then the random part is going to model individual deviation from this mean trajectory. So first of all, the deviation of one individual from the mean trajectory can be due to its genetic additive effect. So this term represents the evolution of the genetic value across environments. The variance of this term is constrained by the genomic or pedigree relationship matrix that we calculated earlier. Also, the deviation of one individual from the mean trajectory can be due to the permanent environmental effect. So this effect regroups all the effects that are specific to an individual, but not from genetic additive origin. So it can be dominance effect, epistatic effect, or microenvironments effect specific to this individual. And at the end, we have the residual with an heterogeneous residual variant for each environment. In this model, these three terms are modeled by trajectories. There are many kinds of basis functions that can be used, and we decide to use two-order Legendre polynomials that allow flexible adjustment and quite fast convergence. So now I'm going to show you some outputs that we can have from this kind of random regression model. So as I explained you in the model, these three terms are modeled by trajectory. So at the end, we will have estimation of variance components, not only for one single point, but for the whole trajectory. So for example, we can represent here the heritability of the ring surface across all environments, across all levels of aridity. So this heritability is estimated with just one regression models. It seems quite constant, even if it tends to increase in most humid environments. The values are quite low, between 0.1 and 0.2, but it's quite consistent and even a little bit higher to what we got with univariate or multi-trait models. And here in this part, you have the genetic correlation between environments. So at each point, you have the value of the genetic correlation between two levels of aridity. So as we can see, we have many environments, many levels of aridity that are really highly positive correlated, but this correlation tends to decrease in driest environments. So it suggests that in this part, we can have a genetic by environment interaction and a re-ranking of genetic value between driest environments and all other environments. And at the end, I also wanted to show you last figure. So it's the evolution of the individual genetic values across environments, estimated genetic value. So it's a representation of this term. So here we have one line for each individual, and it's the evolution of the genetic blood across the environment, the level of aridity. So as we can see, we have a quite important range of variation of genetic value in humid environments, and it becomes more tightened in dry environments. This is pretty nice because you can visualize individual trajectories, but as we have 600 individuals, it can be difficult to visualize and to interpret it. So we proceed to a clustering of trajectories. It gives this kind of results. So it's the same idea. It's a genetic blood across environments. In black, you have the individual trajectories, and in color, it's the groups determined by the clustering. And in here, you have the percentage of individual in each groups. So it helps us to identify general behavior and to typologize trajectories. For example, in the center, we have 3 groups where individuals have genetic value that stay constant over the environment or just slightly increase or slightly decrease. And we have 2 groups, the group E, where we have individuals with high genetic values that tend to increase in most humid environments. And we have at last the group D, with individuals with lower genetic value that tend to increase in driest environments. So we can just modify the previous figure. So it's the same as before. We have still one line per individuals, but we put color corresponding to the group determined by the clustering. So we can see that the clustering mostly applies on the level of genetic values and on the shape of the curve. And we also find a result I previously described. As you can see, in many environments, there are not many crosses between curves, except considering the driest environments. So as I explained with genetic correlation, we have a G by E interaction in this part and a partial re-ranking of genetic value between driest environments and all other environments. So it can modify, for example, breeding choice, breeding strategy, depending if you want individual with quite high genetic value in all environments, or if you want individual with high potential in specific environments. So to conclude, I've shown you how to build normal reaction using densitometric phenotypic measurement, simple climatic characterization, and genetic data in order to visualize individual genetic value across a range of environments. For this part, we have many prospects, but the most important one is the improvement of the environmental characterization. So for the moment, we just use the Arton index with cumulative rainfall and mean temperature, but we plan to improve it with consideration of the previous year and a better differentiation between our 2 locations. Also, we plan to use a growth simulation model called GQE-Le Moulon, which will be based on the environmental characteristic of each site and will allow us to access to more sophisticated parameters like soil water reserves or evapotranspiration demands. And we plan to integrate this parameter in our annual index. And at the end, I only speak here about the ring surface evolution, but we can do the same for ring width or ring mean surface. So thank you for your attention, and I just wanted to thank B4EST project to give me the opportunity to present you these results. B4EST project and University of Bordeaux that funded this PhD project, and the Evolutionary group that funded a part of my trip here to Lisbon. So thank you all.
So, thank you very much, Victor, for your quite nice presentation. Are there any questions? Yes.
Yeah. So it's good because somehow it confirms that the genetic gain is not that good to because we observe in the field. I did not understand how you cope with the young trees, with the young ages, because you were saying it was growing faster. But then you did not talk so much, so I missed that.
Yes, we have. Yeah, in fact, we did not consider the first year because it's, as we can see, it's we have an age effect really important.
So you did not take this.
Yes, we just focus on the mature stage from 2004 to the end.
Other questions?
Why is the
I don't hear you, sorry.
It's only markers.
Sorry?
It's for the matrix you use the result marker.
Yes.
You bring the source marker is all the markers. If you select some of the markers.
So there have been genotypes with B4EST array, but we reduced the number of markers because we have some problems of genotyping. So at the end, we focus on the 300 high quality, 3000 high quality SNP and build the geometrics on. Yes.
I saw maybe you could select those in the magic marker, not just like we have so let's use the array, but then most of the market is not so dramatic. Then it will affect the matrix actually. It cannot capture the means of one market.
Okay.
Other questions? There's still time.
Hi, I was wondering about the time.
I can closer because I don't understand.
In one one, is the time really dependent on the way that you want to model. So it means that you have to have some prior knowledge, I guess, on how it's affecting the training, right?
Yes, actually, for the moment, we just focus on one index symbol, which was easy to model with some just prior knowledge, but we are trying to improve it in integrating, for example, other parameters like soil humidity, or to modify your period of speculation to see which index is better to model and to see the differential response.
It's a very nice presentation. Thank you. I really like how you included the Turing work in this analysis. Basically, regarding the first question, that was the question about the first three rings. I was just wondering whether, if I see this graph with the clear juvenile growth trend, if it would be better to use basic area increment, because in the end we are interested more in production, isn't it?
Yes.
So it's better to transfer the ring widths to base area increments.
Yes, it could be another parameter to study. Okay.
Are there also questions? There's time for one last.
Thank you so much for this nice presentation, Victor. I would like to talk about signal of extreme events. You are able to see also on these trajectories. Do you think that you have signals in your individual deviation, just to imagine some specific response to specific events, because we are in this recording, a nice recording of reaction over time of the trees with this profile.
Yes. So for the moment, we just focus on annual growth regarding annual index. So we sum up the growth on one value, but one prospect of the thesis was to apply normal correction for intra-ring trait, intra-annual variation. So we can model the phenotypic evolution inside a year and regarding environmental or climatic variable measure as a much finer scale. So we can see direct response to some specific growths or in specific months. But for the moment, it's just at the annual risk.
Thank you again, Victor, for your presentation. And now the next one.
Yes, the next one is Mathieu Thierry, who is going to present us some implementation of group selection in forest breeding. Thank you. Probably you need this. You have to speak very close.
Not for shifting, but for the point.
So good morning, everybody. Thanks to the organizers to allow me to share my work here. And we will go deeper in the normal reaction in another kind. And we will go also deeper in the diversity in the genomic prediction. A bit more than Laurence showed us this morning. And this work has been done in a collaboration with the Uppsala University, INRAE of Laurent, and followed by B4EST. So I'd like to spend a bit of time on what we meant by group selection in the context of breeding. In the classic genetic or genomic selection, we would score individual according to its genetic merit, selection index, anything related to breeding values. And we will constitute the population of the next generation based on the best individuals according to these values. So the population would be an aggregate of individuals with the best breeding values, the best scores. But the first observation is that an aggregate of population is not a population for different measurable reasons. To start with, we have the heritability, which is a measure of how much a population can respond to selection. And we select for individual according to their breeding values, and eroding the diversity by doing so, we will inevitably cause the heritability decrease from one generation to the next. We can also talk about the interactions between the individuals that are neglected when we are choosing the individual according to their breeding values. And for the specific case of forestry breedings, once they are planted, they will have the same neighborhood for lifetime. So we would, by neglecting the interaction between the individuals, we would neglect the strongest source of biotic interaction, which is the competition between the conspecifics. We can also talk about the parasites. When we select for individual according to their breeding values, potentially resistant to some pathogens, we would increase the selective potential to pathogens that would promote the selection of certain parasites that could be resistant to the resistance that we do not want. And that is not possible to prevent it if we select for individual according to their breeding values. So there's a lot of reasons to consider selection on a population scale, population levels. So I will show you two different ways to do it. One benefiting from the long-term genetic gain, and one benefiting from the short-term genetic gain. So first off, it is optimal contribution selection. Laurence has mentioned it a bit, but I will go a bit deeper. So it is a method to optimize how many offspring one individual should have based on its breeding values and based on how much the diversity will be eroded if we only take one individual. So it explicitly states that we should balance genetic gain and erosion of diversity. So this here in the equation, it says that we should minimize the population concept, which is a measure of the erosion of diversity, while maximizing for genetic gain, and simultaneously optimizing for both has a cost, comes at a cost, which is summarized here with the parameter alpha, which is the trade-off between the selection and the erosion of diversity that we're willing to make. And so to see the long-term consequence of caring for diversity, penalizing genetic gain by diversity, we have considered the dataset of black poplar developed at the INRAE of the populous Negro. There were 1,000 individuals replicated 8 times in a random block design. There have been phenotype for height, circumference, and resistance to rust. There have been genotype for 7,000 SNPs, and this population is a simple population in a way that there is no population structure whatsoever. And the structure we can see here on the PC of the genotypes, the only structure we can see here is the family structure, where each dot is the individual, they are the offspring of each parent, and the parents are in the label. So each dot are between its two parents, meaning that the only structure we can see here is the family structure. So it is a very simple population. And we have started with this simple population to simulate ahead 20 generations of a breeding scheme, mimicking genomic evaluation and selecting the best individual based on the OCS equation. And we have simulated that for different values of alpha, different trade-offs we were willing to make to see the consequence, the long-term consequence of different breeding strategies. So we were interested in the genetic gain on the long term and the erosion of diversity on the long term. So on the left, you have the genetic gain over time, and on the right, you have the erosion of diversity over time. And each curve is a different value of alpha. I'm not sure we can see the colors, but the more it is red, the less we care about diversity, and the more it is blue, the more we care about diversity. So as we can expect, when we do not penalize genetic gain by diversity, and in the short term at least, genetic gain is the highest. We have the highest return when we don't care about diversity. So the red curves are on top and the blue curves are on the bottom. And likewise, the erosion of diversity skyrockets when we don't care about diversity. So the red curves are on top and the blue curves are on the bottom. So this is what we expect, nothing new in the short term at least. But the thing which is interesting in the long term, and this is the trick here, is that for the red curves, when we do not care about diversity, it will end up reaching a plateau. It will end up saturating. And this is what the so-called selection plateau that Laurence talked about just before. Meaning, again, that selecting only for breeding values at first, not caring for diversity, will make the population lose some of the selective potential that will sooner or later will impede the progress of the population. So this is already a way to include genetic diversity so that we can end up having a long-term return. So for this simulation, they have been they reached the limits with 20 generation, but the basic principle is the same. The less we care about diversity, the lower will be the selection plateau, which means that on the long term we will lose if we do not care about diversity. So jumping to another scale of time, what is the consequence of maintaining diversity on a short term? What is happening in the meantime? So in a forestry breeding, because of weeding unwanted plant, providing good quality of water, soil, and so on, most of the competition is due to intraspecific competition between the planted candidates. And an easy way to say it, to show it, is to consider a focal individual and its neighbors around it. And the question is how the focal individual is affected by its neighbors. And this is a way to include the interaction I have told you about before in the breeding schemes. But of course, if we try to estimate interaction between each pair of genotypes, it would require, and let alone trying to find the genetic basis of it, it would require a huge amount of data. And for now, we have not been very successful for doing so. As we can see on this graph, you have the prediction, the predict phenotype on the x-axis, and the observed phenotype on the y-axis. So the circle is when we not took account of interaction. It's the process is when we take account of the interaction. As you can see, the predictive ability is pretty good, but there is no difference whatsoever between the circle and the crosses. They are completely overlapped, meaning that we did not have enough power to estimate what the interaction would provide to the breeding. So we have to find some way, a more simple way to include the interaction between in the breeding scheme. So what about considering that the phenotype would depend on the local ancestry? What about the local ancestry is how much a focal individual is related to its neighbors? And it would make sense in an evolutionary framework in which if there is kin selection, for instance, competition will be weaker between related individuals, and on the opposite, if there is niche competition, competition will be higher when individuals are related. So there is reason to believe that phenotype would depend on ancestry that I have noted theta n and for neighborhood. So we will go for that. And indeed, it is what we see on the first glance when we plot the height of the focal individual as a function of the local ancestry. The colors are the different families. Well, it might not be like a potato here, but each slope is significant, meaning that indeed the phenotype of the focal individual depends on the local ancestry. But since everything is linked to everything in this framework, you could unfortunately see the same kind of pattern when we plot the height of the focal individual versus the global mean ancestry, which is how much an individual depends on every other individual of the population. So we were not sure what we were seeing here, if it is something that the focal individual depends on the local ancestry, kin selection, niche selection, which would reflect past selection on relatedness, or if the phenotype of the focal individual just were not affected by local ancestry or local effects, and reflect past selection on related individual, because past selection has selected related individuals. So how to disentangle those? For that, we have used the same genotype, the same dataset than before, the dataset of the black poplar. But this time we have used it mainly because it provides a very interesting block design, which is the random block design, meaning that if there is any local effect, it will be unrelated to local ancestry because we have randomized it. And any local effect will be ascribed can be ascribed at least, to micro-environmental effects. And this is what we have done, and this is something that we have tested. This is pretty accurate. When we plot the local ancestry versus the global ancestry, each dot is an individual. It follows a slope 1-to-1, meaning that the local scale is a good reflection of the global scale, meaning that the randomization was pretty efficient by doing so. So we will go for it. We will assume it. And for that, we will fit not a classic animal model, but a slightly modified one. For the phenotype Y, we will fit a spatial effect. We will account for the smooth local micro-environmental effects that will absorb all the local effect that is not local ancestry, because local ancestry cannot be smooth because we have randomized it. There is also the animal model of the polygenic effects, animal effects, sorry, that will absorb the breeding values, and a residual E following a Gaussian distribution that we will refit to be a function of the local ancestry, theta n, plus a residual. All that to account to measure for each genotype its plasticity, its reaction norm to local ancestry. And this is only possible because the block design is random, and because we have replicate for each genotype. And we have measured here the ancestry, the local ancestry with the pedigree values, with the matrix A, or the genomic data with H, and they are on the same scale. And similarly to what Victor has shown you just before, the values of the matrix A, the relatedness computed with pedigree, is very close to what is computed with the genomic data. And just that the genomic data provide much more amplitude, much more precision. So we will see what we will obtain. So this is the final graph of the presentation, and each dot is one genotype, one individual. On the x-axis, you have the plasticity on local ancestry computed with the pedigree. And on the y-axis, you have the plasticity of each individual computed with the genomic data. The first thing you can see that since we're refitting residual, it is all the values are centered around 0, which is pretty normal. So meaning that some of the individual will grow taller if we consider height when they are surrounded by related individual, and some individual will grow slower compared to the population mean when they are surrounded by related individuals. The second thing that we can see is that plasticity is a bit higher when computed with pedigree than with genomic data, meaning that the phenotype of a focal individual would much more depend on a relatedness when computed for local ancestry computed with the pedigree than computed with genomic data, meaning that the focal phenotype is more sensible to how close or how far the individual is in terms of pedigree than in terms of genotypes. So it can be an artifact because the values of air, the range of values of air are pretty rough, or it can reflect something biological that I cannot know yet. One of the hypotheses is that the genomic data is more precise, but might be more biased to capture some of the co-selection of what I have told you about just before, convergent selection and so on, that might be an impediment when we want to measure what is related to gene selection, niche competition, and so on. The third thing that we can see, and this is the solution of the main problem that we had, is that this framework, when we try to predict values and compare it with the observed value, the predictability, accounting for the plasticity to diversity, increase the predictability by 4 points. It's not 4%, it's 4 points, which is not negligible. meaning that we have solved the first issue that we had, that we wanted something measurable and usable for breeding schemes, so that we have now. So to conclude, I have shown you different ways to include the genetic diversity in a breeding scheme. There is a way to higher the long-term diversity by maintaining genetic gain, and meaning that we're just sustaining a selective potential of a population that will end up in a higher long-term genetic gain, or in a short term by optimizing the interactions in a simple way using the local genetic diversity. So with this, I'd like to thank the University of Uppsala, the collaborators with the INRAE of Orléans, and B4EST founding me, and my supervisors, of course, Leopoldo Sanchez and Martin. And thank you very much.
We have time for some questions.
I have a question regarding this co-ancestry or local co-ancestry. Have you an idea of the different kind of field or breeding testing? We have in breeding different test options. We have a full seed progeny test, we have accident, and with different randomization, possible randomization. Do you think that from your experience, we can imagine to have change in the way we are doing this field evaluation in order to make to better make value of this local co-ancestry expression in the phenotypic performance? How could we learn from that? Maybe to have change in the way to do testing.
So to answer your question, the best kind of data and the test that we could implement is, again, the diversity. That's everything, the statistical power that we have comes from the fact that we have a lot of different local co-ancestry that each genotype will experience. So for now, we have used a data set that is not meant for it. We were lucky that the block design were randomized, that the individual faced a different kind of local ancestry, and we didn't use it as its best potential. But if we optimize this, providing different kind of interaction in the data set would be the best to take advantage of this framework and predict the genetic gain in the long term and the short term using this.
No more questions? Okay, so I think it's time for the break. So please be back at 11 for the next presentation. And well, thank you very much, Matthew. La personne qui parle. Comme les gens y parlent de ce côté-là, peut-être qu'on peut mettre un micro comme ça de ce côté-là. Comment là, on en est là. Comme ça, on peut utiliser ça pour la question. Voilà, on peut le mettre là comme ça. Très bien.
Peut-être pour les présentations, quand tu parles sur 20 minutes, on peut le conserver le niveau après, il parle ensuite à la fin, il se met ici, mais oui, ça c'est plus facile. Ça va? Et donc, c'était
Quand ils n'ont pas collé sur la bouche, il faut vraiment l'avoir collé sur la bouche. Mais si quelqu'un se connecte à distance, non, mais si. Ouais, je vais passer. Là, actuellement, ce qu'on fait, c'est qu'on filme la caméra, on filme l'écran.
On enregistre.